Hello, how are you doing? Welcome back. In this video, we will be like for this chapter, the DNA chapter, we'll be dividing these videos on to separate videos, or I'll be trying to put them in one, but I do not promise. First, teach or explain, and then revise, and then revise, and then revise. So basically, I'll tell you in case you want to do it alone, if you like studying by reading or whatever. I'm going to teach you from these notes, from the PMT notes, and then after that, we were going to revise from this Notion link. After that, we will be doing some, just like, it's not really to revision, because this is for Igcse. Here, not this one, where is it? Yes, it's not really, and it doesn't really have lots of stuff about, like, for example, it doesn't have DNA here, but it has some stuff. So basically, sometimes we can go for photosynthesis, for example. It's just going to be one page that has some notes and stuff, just like revise for that. I'm going to put you all these links in the description below. After that, we might be doing some Quizlet. Before we do, like, before the Quizlet, or after the Quizlet? Yeah, after the Quizlet. Like, we'll do the Quizlet, and then we will be doing the key points. Before the key points, we'll be doing the finishes, but key points are very important, still from the PMT thing. So this is basically how to study, a way for studying, if you would like to say it like that. You can also cover some stuff from the syllabus if you'd like to, like, go through the stuff from the syllabus. But until we do so, let's start with the DNA and meiosis chapter. First of all, there's something called nucleotide. What is a nucleotide? A nucleotide is a section of DNA made up of sugar, phosphate, and a base. It's not written, I'm telling you, is it written here? It's not written directly, but it's written that it's basically, nucleotide is a sugar called deoxyribose and a phosphate group, and an organic base belonging to one of the two groups, either a single ring cysteine and thymine or a double ring adenine and guanine. Whatever it is, basically a nucleotide is a sugar. One second. Is a sugar, phosphate, and organic base. Organic base has two things, either a single ring cysteine and thymine. Cysteine and thymine, basically the C and the T, or adenine and guanine, which is basically the A and the G. And the single is cysteine and thymine. Set is single. You can see over here. You can see. How can you remember? Let me tell you. So basically, we have the letter C, and then we have the letter T, right? These are single. Like, this is only one line, and this is only one line. So basically, this is single, one line. For the T, for the double bond, you say line, second line, third line, and then the other one, A and G, line, 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 and line. So basically, lots of lines, double bond. This has two, so do not try to say two. So just say, like, lots of lines is the biggest, so the two, and the small little ones are the one single bonded. And after that, let's talk more about it. The deoxyribose sugar, phosphate group, and organic bases are a result of condensation reactions to give a mononucleotide. Two nucleotides combine to form a dinucleotide. The continuous of mononucleotides forms a long chain known as polynucleotide. Let's just go back in this part, the part of the biological molecules. Okay, you know what a mono means? Mono means one. The word mono itself. Okay, the word mono itself means one. The word di means two, the prefix, and the word poly means more than two or three, more than three, right? So, a mononucleotide is one single base unit. When you add them together by a condensation reaction, which is the removal of water, and the products are the product itself plus water. When you put two mononucleotides together, they form a dinucleotide. More, they make a long chain called polynucleotide. The nucleotide itself is the monomer of the polynucleotide, okay? which is of the polymer itself. So let's go. There is something called the DNA structure, which we must know about it. The DNA structure, basically what we need to know about it, that in 1953, Watson and Crick worked out the structure of the DNA. DNA is basically made of 2 strings of nucleotide, each of which is extremely long, and they are joined by hydrogen bonds formed between bases. I know this might sound easy, and it is easy, but we need to focus on some stuff. From that they are joined by hydrogen bonds, first important point, and that they are formed between the bases. And in Oxford, they really love the word bases. Like whenever you have the chance of putting bases, just put it. Bases is very important. We also need to know that it's made of 2 strings. We might need to know that it's extremely long, and we need to know that it's made of nucleotides. And we need to know the name of all this, which is basically the DNA. Because I know people who really like, and I'm one of them. I remember all this stuff, I remember, but I do not remember what was it. So you need to make sure that you know that all of this is basically the DNA. So let's just revise a bit. What is a DNA? A DNA is made of 2 strings of nucleotides, each of is extremely long. They are joined by what? By hydrogen bonds. Formed between what? Formed between the bases. Okay, so do you know anything about the bases? Like, did you ever hear about the bases? I did. And we will be talking about the pairing of bases. Okay, so basically, you know that there is something called complementary bases. If you do not know what a complementary base is, it's very important to know. And it's mentioned over here. Complementary bases is basically that a adenine always pair with thymine, and guanine always pair with cytosine. This is the complementary base. But that was the part of the O level where we said A at the garage, and we said that the C looks like the G, so it's the same thing, and then at looks the same. What was it? But this was for the O level part. Now for the A levels part, you need to know that the A is adenine, the T is the thymine, the G is the guanine, and the C is the cytosine, and these are called complementary bases. But is that only the thing? No, it's not only the thing. What you need to know about adenine and thymine as well is that they always pair with 2 hydrogen bonds. And this is very important. Do not try to skip it. It's very important. Second thing you need to know about guanine and cytosine is that they form by 3 hydrogen bonds. How can you remember that, mate? Okay, you say adenine and thymine. First line here, second line here. So 2 hydrogen bonds. And then you say cytosine with guanine, which is basically 1 line, second line, third line. So 3 hydrogen bonds. Okay. Then we go to the third important thing. That we need to know that the pairs of bases, the organic bases contain basically nitrogen and are 2 of types. Those with a double ring structure have longer molecules than with those with a single ring structure. This has lots of information and we need to color a lot. So, the organic bases contain what? Contain nitrogen. So, you need to know that they contain nitrogen and there are 2 types. This is the first important thing. What is the second important thing? That we need to know that those with a double ring structure have longer molecules than those with a single ring structure. So, as you can see over here, it's not shown over here. Sorry. Single ring structure, as we said above, right? And double ring structure. Single cell was adenine, thymine, and basically they are shorter than the guanine and the adenine. Again, thymine, cytosine are single rings, and they are shorter than adenine and guanine, which are basically longer. And the last thing is basically adenine is said to be complementary to thymine and guanine is said to be complementary to cytosine, which is basically always pairs. Okay, is that easy for you? Let's repeat again in case you forgot. Oh, one of the things that I forgot mentioning of what we study is that we write notes. I study first the notes. I always study the notes, these PMT notes, and then go write my own notes. I post them on the Instagram, do not worry. But try your best to study the notes I'll write and write your own notes as well. Okay, let's go. Pairing of bases. Basically, the organic bases contain nitrogen. The what? The organic. Okay. The organic bases contain nitrogen and are of two types. Those with a double ring structure have longer molecules than those with a single ring structure. Adenine always pair with thymine by 2 hydrogen bonds. Guanine always pair with cytosine to 3 hydrogen bonds. Adenine is said to be complementary to thymine and guanine is said to be complementary to cytosine. That's great. But I know we've been talking a lot about DNA. From the O levels to now here the A levels, going through everything. It's basically like it's everywhere. What is the function of it? Why is everyone saying it's important? We always hear of a gene, allele, chromosome. What is even that? Let's go. DNA basically has lots of functions, where DNA is adapted to carry out its functions in numbers of ways. First of all, it's very stable and can pass 2 generations without change. Number 2, its 2 separate strands are joined only with hydrogen bonds which allow them to separate during replication. Number 3, it's a large molecule and carries an immense amount of information. 4, by having the base pairs in the helical cylinder, the information is protected from being corrupted. These are basically the adaptations of the DNA. But let me ask you another question. Do you know what are the functions of the DNA? Simply put, it contains the instructions needed for an organism to develop, survive, and produce. Okay? And to carry out these functions, to the human being to carry out developing, surviving, and reproducing, DNA sequences must be converted into messages. We will be talking about later the message part, but for this part, we need to know that there are some adaptations to maintain these functions, surviving, developing, reproducing, etc., right? So what are the adaptations again? Let's again color. But in case you do not like my colors, write me in the comments what colors you love. Do you like these colors? Me, myself, I do not, but why not try them? Let's get started. So, the function of the DNA. So basically, DNA is adapted to carry out its functions in a number of ways. Number 1, it's very stable and can pass to generations without change. So it's very stable and it can pass to generations without change. Say after me. DNA is adapted by being very stable and can be passed to generations without change. Number 2, which is very important as well. Its 2 separate strands are joined only with hydrogen bonds. Okay, why is that important? To allow them to separate during replication. I get it. What about this point? Is it important? It's very important. What is this point? It's a large molecule and carries an immense amount of information. Okay, last important thing. By having the base pairs in the helical cylinder, the information is protected from being corrupted. Right. So basically, let me just tell you something. DNA itself is very stable and it's a large molecule. Being very stable and being a large molecule makes this DNA pass to generations without change and carries an immense amount of information. Great. Then after that, we get to say that the structure of it has 2 separate. No, not this color. Sorry, guys. The structure of it itself has 2 separate strands. Okay, and also have the base pairs in the helical cylinder. Great. What's after is that we say that they're only joined by the hydrogen bonds, and here in the information is being protected, which allow what? Like, what is it? Allow them to separate during replication from being corrupted. Let's read each color alone. DNA is very stable and it's a large molecule. DNA can pass to generations without change, and DNA can carry an immense amount of information. DNA has 2 separate strands, and DNA have the base pairs in the helical cylinder. DNA is only joined with hydrogen bond and hydrogen bond, which allow them to separate during replication. And here, by having the base information, is protected from being corrupted. This is actually very important. I'll save it and make sure to write it this way in the notes. This is very important, guys. No, this is actually very important. Okay, now we go to the triplet code. Before we go to the triplet code, we'll go to the triplet code. What is a gene? Before we go to what is a gene, do you know what is a gene? Do you remember what is a gene? The definition itself, like, not really talking. Okay, the gene itself is a section of a DNA that codes for making a polypeptide. Genes are sections of DNA that contain the coded info for making polypeptides. Right. The coded information is in the form of a specific sequence of bases. Polypeptides combine to make proteins, and so genes determine the proteins of an organism, and as enzymes control chemical reactions, they are responsible for an organism's development and activities. Let's repeat that again. Let's do it. What is a gene? Genes are sections. Okay, so genes are what? Genes are sections of what? Sections of the DNA that contain what? That contain coded info for what? For making polypeptides. That's great. The coded information itself here, the coded information is in the form of specific sequence of bases. So basically, genes, which are sections of DNA that contain the coded info, contain form of specific sequence of bases to make polypeptides. Polypeptides, which are made of the specific sequence of bases that are coded into the sections of the DNA, which are called genes, combine to make proteins. Combine to make what? To make proteins. Polypeptides combine to make proteins, and so the genes determine the proteins of an organism. That's why it's important for development, survival, and reproduction. And as enzymes are basically proteins, control chemical reactions. They are responsible for an organism's development and activities. So that's one of the main functions of the DNA. Would you like to repeat it with another color, or let's just use the same another color? But again, what is a gene? Genes are sections of the DNA that contain the coded info for making polypeptides. So the coded info is in the form of specific sequence of bases. So the coded information is basically specific sequence bases. So the genes are sections of DNA that contain a specific sequence of bases for making polypeptides. The polypeptides made of the genes, which are basically section of the DNA that are made of the coded info. The polypeptide itself is basically combine to make proteins, and so the genes determine the proteins of an organism. Enzymes, which are basically proteins, the gene determine the proteins of an organism, control the chemical reaction. So here the genes determine the protein, enzymes control the chemical reaction responsible for organism's development and activities. Great. Now we go to something that's called the triplet code. What is a triplet code? So before we're actually doing that. So the triplet code, basically, for your information, scientists suggested that there must be a minimum of 3 bases that code for each amino acid. So what is a triplet code? 3 bases that code for each amino acid. But let's just use another color for this part so we can memorize it by this color. Do you think better, blue, yellow, orange, or yellow, green? Not sure. We can use this color or the orange color. We'll see. Okay, so scientists suggested that there must be a minimum of 3 bases that code for each amino acid. But do you know how many amino acids is there in the whole entire world? There are only 20 amino acids. But they have millions of other shapes, sequence bases, etc. That's why there are lots of types of proteins. Lots, for real. Each amino acid must have its own code of bases, right? And only 4 bases are present in the DNA. A, T, C, and G, right? If each base coded for a different amino acid, only 4 amino acids could be coded for. Using a pair of bases, 16 codes are possible. 3 bases produce 64 codes. So yes, there are only 20 amino acids, and each amino acid must have its own code of bases. Only 4 is used, okay, a percent, but using a pair of bases, 16 are codes are possible, and 3 bases produce 64 codes. That's good. Okay? Let's just repeat again and say that the triplet code only, amino acids are only 20. And each amino acid must have its own code of bases. And that there are only 4 in the DNA. And if each base code, that's a mathematical thing, and if each base coded for a different amino acid, only 4 amino acids could be coded for using a pair of bases. 16 codes are pair of bases, only pair, right? 16 could be possible, 3 bases, 64. Right. Further experiments have revealed the following features of the triplet code. First of all, a few amino acids have only a single triplet code. Number 2, the remaining amino acids have 2 to 6 triplet codes. Wow. Number 3, the code is known as a degenerate code. 4, the triplet code is always read in one direction. 5, the start of a sequence is always the same triplet code. 6, 3 triplet codes do not code for amino acid and are known as top codes. 6, I think, the code is non-overlapping. 7, the code is universal. I know I said the code a lot this time, so let me count how much I said a code. Code as a here, code another one here, here, 3 more times in the same sentence. Here's another 2. Can you see how much important it is? So let's repeat again using another color to remember them. Blue, maybe, I guess. Okay. So triplet code. There are some information about it. First of all, a few amino acids have only single triplet code. The remaining amino acids have 2 to 6 triplet codes each. The code is known as a degenerate code. The triplet code is always read in one direction. The start of a sequence is always the same triplet code. 3 triplets codes do not code for an amino acid and are known as top codes. 3 triplet codes are known as top codes. Remember, the code is non-overlapping. The code is universal. Great. DNA and chromosomes. Before we just talk about the prokaryotic and eukaryotic cells, do you know what a chromosome is? Chromosome is a thread-like structure made of protein and DNA, passes hereditary info on. Basically, in this chapter, we're gonna keep on saying DNA. So, just like DNA. Okay, so DNA and chromosomes. I'm not sure if I can highlight again the DNA to tell you how important DNA is here in life, but I see you can see the letter DNA and A. It's very long. Anyways, chromosome again is a thread-like structure made of protein and DNA. Passes hereditary info on. And then there are the prokaryotic cells, such as bacteria, and the eukaryotic cells, which basically not like bacteria, much larger. In prokaryotic cells, such as bacteria, the DNA molecules are smaller. They form a circle and are not associated with protein molecules. They therefore do not have chromosomes. Ooh, skip this part. Not skip it, guys. Prokaryotic do not have chromosomes. That's the thing. And DNA here is smaller and they form a circle and are not associated with protein molecules. Basically, this sentence is very important. In prokaryotic cells, such as bacteria. Bacteria is an example of a prokaryotic cell. There are 2 types of cells, if you know, right? And we're going to do another chapter about cells. Prokaryotic cells, such as bacteria, the DNA molecules are smaller. So, in prokaryotic cells, DNA are small and they form a circle and are not associated with protein molecules and they therefore do not have chromosomes. Now, what have chromosomes? My lovely eukaryotic cell. The eukaryotic cells, the DNA are larger. They form a line, not a circle, and occur in association with proteins, just the opposite prokaryotic, and they form chromosomes. Do you know what a chromosome is? A chromosome, basically, as we said, is a thread-like structure made of protein and DNA, passes hereditary info on. Like, what structure does the lovely chromosome have? Basically, it's only visible as distinct structure when a cell is dividing. The rest of the time, they are widely dispersed through the nucleus. Number 2, when visible, they appear as 2 threads joined at a single point. 3, each thread is called a chromatid. 4, the DNA in chromosomes is held in positions by proteins. 5, the length of a DNA is highly coiled and folded. Basically, the DNA is coiled and then looped and further coiled before being packed into the chromosome. The number of chromosomes varies from one species to another. But before we go to this part, let me just tell you more about the chromosome structure. I don't know why I'm just like, I feel the chromosomes are basically my enemies for some reasons, but it's not. They're actually very nice. They're not. Anyways, chromosome structure. Basically, they are basically always like dispersed through the nucleus. Okay? But when the cell is dividing, they become visible. Okay? When they become visible, they appear as two threads joined at a single point. Where they are joined, not the word joined. Each thread of them is basically called a chromatid, and the DNA in chromosomes is held in positions by proteins. Here, the chromosome has proteins. The proteins are held by DNA. The DNA is held by proteins, and the length of the DNA is highly coiled and folded, and whatever. This is the DNA. Here are the proteins that hold the DNA. The genes, DNA and proteins are multifolded. And then here's the chromosomes. Each line is basically a single point. Each line is called a chromatid. They look like each other, basically homologous. If they look different, they are heterozygous. No, homologous, heterozygous, you know that. But these are mainly into the genes itself. But whatever. DNA is coiled and the looped and further coiled before being packed into the chromosomes. And the number of chromosomes varies from one species to another. For example, in humans, we have 46 chromosomes, whereas in potato plants, we have 78. And in almost all species, there is an even number of chromosomes in the cells of adults. This is because they occur as homologous pairs. Homologous chromosomes. Sexually produced organisms are the result of the fusion of sperm and egg. One of each pair is derived from the chromosomes provided by the mother and the other derived from the chromosomes provided by the father. These are known as homologous pairs, and the total number is referred to as the diploid number. During meiosis, halving the number of chromosomes is done in a manner which ensures each daughter cell receives one chromosome from each homologous pair. Okay, is everything great over here for homologous chromosomes? If not, do you know what a homologous chromosome itself is? Homologous chromosome, basically two chromosomes determining the same characteristics, one from mother and one from father. This is the only thing we need to know about it. Chromosomes determining the same characteristics, one from mother, one from father. But how can this happen? Yeah, how is that even possible? One from father, one from mother. Basically, they get derived, right? How is that? Again, sexually produced organisms are the result of the fusion of sperm and egg. One of each pair is derived from the chromosomes provided by the mother, and the other is derived from the father. And this is how it comes. These are known as homologous pairs. That's it. And the total number is referred as the diploid number type. During meiosis, having the number of chromosomes is done in a manner which ensures each daughter, because meiosis have the number, because like 46 and 46, you're making a potato and you're not making a human being. So 46 is basically 23, right? Type. What is an allele? Allele. Basically, it was one of the different forms of a gene is an allele. So each gene exists of two or occasionally more different forms. Each of these forms is called an allele. Each individual inherits one from each parent. Any differences in the base sequence of an allele may result in a different sequence of amino acids being coded for. Great. What about meiosis and the genetic variation? The division of the nucleus of cells occur in two ways, either mitosis or meiosis. One of them form diploid cells, and one of them form haploid cells. One of them is used usually, one of them is only used for the gametes, etc. Let's talk about that. Mitosis produces two daughter nuclei with the same number of chromosomes as the parent cells and as each other. Meiosis produces 4 daughter nuclei, which with half the number of chromosomes as the parent cells. Why is meiosis necessary? In sexual reproduction, two gametes fuse to give rise to new offspring. If each gamete has a full set of chromosomes, as I said, 46 and 46, you're making a human, you're making a dinosaur, then the cell they produce has double the number. This doubling of the number of chromosomes would continue at each generation. And in order to maintain a constant number of chromosomes in the adult, the number must be halved at some age. And that's what we use by the meiosis. So meiosis produces 4 daughter cells, each with half the number. Okay? Right. How can meiosis occur? Right. Meiosis involves 2 nuclear divisions that normally occur one first after the other. In the first division, as you can see in the picture, the homologous chromosomes pair up and their chromatids wrap around each other. Equivalent positions of these chromatids may be exchanged in a process called crossing over. By the end of this stage, the homologous pairs have separated this one chromosome from each pair, going into one of the 2 daughter cells. In the second meiotic division, the chromatids move apart. At the end, 4 cells have been formed. I do not expect you to understand it from the first point without knowing what a crossing over means. Crossing over is basically the process where chromatids break and rejoin with their homologous chromosomes to exchange alleles. Before we complete, let me tell you meiosis. Meiosis basically cell division that produces 4 haploid, half the chromosomes, genetically different daughter cells. There is also the independent segregation, which we're going to cover later. And then we have the crossing over, where the process where chromatids break and rejoin with their homologous chromosome to exchange alleles. Okay, since that's the case, then here in the first division, the homologous chromosomes pair up, and the chromatids, single line, wrap around each other. Equivalent portions of these chromatids may be exchanged in a process called crossing overs. By the end of this stage, the homologous pairs have separated this one chromosome from each pair, going into one of the 2 daughter cells. And in the second meiotic division, the chromatids move apart. At the end, 4 cells have been formed, 1, 2, 3, 4 daughter cells, which are all genetically different. Like independent segregation. Independent segregation, basically, definition of it is that homologous pairs randomly separate during meiosis 1 into separate cells. So, during meiosis 1, each chromosome lines up along its homologous partner. When these homologous pairs arrange themselves in line, they do it randomly. Each one of the pair goes into the daughter cell, and with which one of the pairs depends on how they are lined up. Since they are lined up randomly, the combination of chromosomes that goes into the daughter cell is also random. That's why they're genetically different, and this is called independent segregation. Independent segregation, again, homologous pairs randomly separate during meiosis 1 into separate cells. How this happened? During meiosis 1, each chromosome lines up along its homologous partner. When these homologous pairs arrange themselves in line, they do it randomly. Which one of the pair goes into the daughter cell and with which one of the pairs depend on how they are lined up. Since they are lined up randomly, the combination of chromosomes that goes into the daughter cell is also random. This is called independent segregation. Last thing before we finish, crossing over. The chromatids of each pair twist around each other. During this tensions are created and portions break off. These portions rejoin with chromatids of its homologous partner. The equivalent portions are exchanged, and new genetic combination is produced. Again. What happens at crossing over? Or what's that process? The chromatids of each pair twist around each other. Hell, we understand that, right? But during when the chromatids are twisting around each other, tensions are created and the portions break off. The portions that broke then rejoin with chromatids of its homologous partner. خلاص? The equivalent portions are then exchanged, and new genetic combination is formed. Great. That's not for this video. I'm doing the other stuff in another video for like like for what is it called, completion or whatever. Before I finish, I want us like to basically do some make sure you understand all the definitions. Here they are. If you need or ask any question, you can ask me in the comments in the Instagram account. I barely use it, so like better to in the Telegram or whatever you would prefer. I'll try to be active on the Instagram this account. Yeah. Okay. So whatever you would want, I'm here for you. Have a nice, beautiful day. And if you understood at least 60% of this chapter, or 70%, 60%, it's fine. Make sure to be proud of yourself. Okay. Have a nice day.